for the day, with the ECoG mapping, seizure AI, for example, is interesting because this is a project where we look at epilepsy monitoring. And today will also be a lecture about the high-frequency oscillations, which are a very nice biomarker to identify the sources of epilepsy. So all these projects have to do with brain-computer interface technology, and many of them are invasive, so that we can further research on high gamma mapping, CSAPs, high-frequency oscillations, and much more. So here you can see a typical situation in a surgery room. The neurosurgeon has an ECoG strip in his hands, which has 8 channels. On the table, you can see the cortiQ system. On the right-hand side is, for example, the electrical cortical stimulator. And just behind is the patient prepared for the neurosurgery. And for that, you need, of course, also a biosignal amplifier to amplify the ECoG signals. And then you are already prepared for doing high gamma mapping. And this also extends into neuromodulation. So the difference in neuromodulation is that we're also measuring from the brain. Then on demand, in real time, different brain regions are stimulated. So this can be, for example, the thalamus in Parkinson's disease patients. I already showed you this figure a couple of times. So this is Gerd Purtscheller, my PhD supervisor, while I was at the University of Technology in Graz 25 years ago. And he figured out if a person imagines a left-hand movement, then the contralateral hemisphere is activated. So this red spot is called event-related desynchronization. And this is something that we can measure with the EEG. And the big advantage is that we know exactly where we should place the electrodes to capture left and right hand movement. So this follows the international 1020 electrode system. And if we place the electrodes around C3, we get right hand movement. If we place it around C4, we get left hand movement. So it's very easy to know where we should place the electrodes to capture motor activities. But this is not so easy for implants. And this is demonstrated here in a slide from Kai Miller, who also gave a very nice keynote lecture. He's now at Mayo Clinic, and he implanted 64 ECoG channels in one epilepsy patient. So here you can see these ECoG grids. Every little white dot is one platinum-made electrode. They have a distance of 1 centimeter. And we have one grid here with 32 channels, another grid here with also 32 channels. And then basically 64 channels plus grounding cable are coming out of the scalp of the patient, and they are connected with the brain-computer interface. And Kai first asked the patient to be at rest, not to do anything, and he calculated the power spectrum that you can see in blue. So we can see the characteristic 1/f damping of the EEG. This means with higher frequency, the energy gets less. And if you look at the hand movement, so afterwards Kai instructed the person to move the hand, and he calculated again the power spectrum, and you can see here the red line. And interesting is, of course, to compare how large this is extending. And so the blue line here in the low frequency band is, for example, during resting, and the red line is during the hand movement. You can see during resting, we have more energy in the low frequency band. So this is what we already know as event-related desynchronization. And you can see there's a pretty wide cortical network involved in this activity. And that's the reason why we can also measure it with EEG electrodes on the surface of the head. But if we are going to higher frequencies, so from 80 to 100 Hertz here, you can see in the high frequency band, it's exactly the opposite. So in the resting state, we have less energy than doing the hand movement. And this is considered to be high gamma activity, or task-related high gamma activity, because it occurs as soon as you're doing the hand movement. And if you look at the cortical network, then you can see that one single electrode is coding this information. And this means the resolution, the spatial resolution, is just very high. But this produces another problem. So you really have to put the electrode over the cortical network coding the information, and otherwise you wouldn't find it. So if you would analyze the electrode in the neighbors surrounding here, you wouldn't get the hand movement anymore. So finding electrode positions is very important for ECoG recordings, much easier for EEG. And then Kai put a data glove onto the epilepsy patient, and he was asking him to move 6 times the thumb. You can see it's going up and down 6 times, and then the patient is at rest, and then he is moving again, rest again. And then he took one single electrode. So in this case, the dark blue electrode, which is located here over the sensory motor cortex. So this is the thumb position, and he decoded with the brain-computer interface the movement indicated here in purple. You can see, we can somehow see the number of repetitions. In between, we don't have false positives. And very interesting is this overshot here. So when the person is doing the first time the movement, we get a big overshot. There's a lot of high gamma activation occurring because it's a new task for the patient. And then with every single repetition, it gets smaller and smaller because you don't need your cortex anymore. The spinal cord takes over. And this is very important information for designing brain-computer interface experiments. So if you're doing repetitive finger movements, you don't need your cortex anymore. Same is the case for the index finger. So you can see he moved 7, 8 times the index finger, and we get again the high gamma overshot when he's doing it the first time, and then it diminishes out. We don't have false positives, and here we see it again. And the same is also the case for the little finger. So we see very nicely here, for example, the number of little finger movements. And this is outstanding because this type of resolution, so single finger movement resolution, is really difficult to get with non-invasive recordings, but with ECoG recordings, we have enough spatial resolution to get this perfect picture here that Kai produced. Then we are here in this image, you can actually see how such an ECoG grid looks like. So every single electrode is made of platinum. Then you can see a wire going to the cable, and these 2 cables are actually coming out of the scalp, and they are connected to the biosignal amplifier. You can see the distance is 1 centimeter, center to center, and this defines actually the spatial resolution of a standard clinical grid. Such a 64-channel grid costs around 1,000 euros. So they are inserted into a patient, they do one recording, and then it's thrown away. So they don't reuse it because it's risky if you use it for another patient. And here I have a couple of videos so that you can see how the ECoG brain-computer interface research developed. So this from Gerwin Schalk. And in this case, they tried just to move a cursor into target positions. So this was done 2004. You can actually see how nicely this works. So this was one of the first experiment where 2-dimensional cursor control with ECoG was realized, at this time with G.USB amp or a 16-channel biosignal amplifier. So this was the first unit actually who could do the real-time decoding of the ECoG signals for BCI interfacing. And then Gerv developed also BCI in 2000, and he hooked it up with Doom. Maybe some people still remember this game. On the left-hand side, you can actually see the ECoG recording. It looks basically like EEG. It's just much bigger, and it has less artifacts because you don't get this eye movement or muscle movement contamination. Then you can see that he is able to control the firing of the gun. So in this case, he is using a joystick. You can also see it in the video to move around in the surrounding, and he can trigger with the ECoG signal the gun firing. So Gerv showed the video also to the US Army, and of course they were very impressed, and afterwards he got very nice research funding from the US Army. So this is about 20 years ago. This is a recording that we did together with Dr. Kamada at Ashikawa Medical University, and also Dr. Kuni, who is at the University of Tokyo. And it was one of our first ECoG experiments we did also 20 years ago. You can see we used MATLAB and Simulink for doing all the real-time processing. On the left-hand side, we are reading in the data from 4 USB amps. You can see they are stacked above each other. They can be used for invasive recordings. And here you can also see the cables coming out of the epilepsy patient. And in front of him, we have instructions like making a fist, making fingers straight, showing the peace sign. On the right-hand side is the cortiQ system recording everything in MATLAB and Simulink. And then you can see here also the power spectrum while the person is at rest and while the person is doing hand movements. That's the classification accuracy. After the instruction, you can see how the error rate goes down. And then we are reaching a certain accuracy. So this was done 2011, so 13 years ago. Was one of the first experiments ever done with ECoG and BCI. This is coming from the Verre project. So in this case, it was a telepresence experiment. So we tried to control a robotic system from Abdel Keda, which has a very nice robotics lab in Japan. And the epilepsy patient from Asaikawa Medical University was actually controlling the ECoG-based brain-computer interface. Now you can see that he's able to lift the robotic arm of this human-like robotic system. So the robot costs about 1 million euros. And the idea here was actually to give a robotic body back to a person who is paralyzed so that he can do tasks for him. So this worked actually very nicely. But of course, it doesn't make a big difference if the robotic system is in the same room as you. We just want to demonstrate that you can also send a robotic body to remote locations to do tasks for you. This is another interesting experiment because, as you know, flickering lights can induce epilepsy and seizures. And for that reason, it was always problematic to run steady-state visual evoked potential experiments with epilepsy patients. And Christoph Capella, doing his PhD thesis with g.tec, developed a code-based visual evoked potential experiment. This is actually the first time that we used CVAPs with an epilepsy patient. And the nice thing is that these little boxes are not flickering with a constant frequency. It's code modulated or frequency modulated. And for that, the likelihood to induce a seizure is just not existing. And this allowed us to run SSVP, or in this case CVAP experiments, also with epilepsy patients, and to look in the ECoG recordings what we can achieve. And this turned out actually to work very nice. And also in this recording situation, you can see the cables are coming out of the head of the patient, and they are connected to the G-high amp. In this case, which is our 256 channel biosignal amplifier, which was developed a little bit later after G-USB amp. This is actually a recording of the data set that we had in the hackathon. So this patient here did different hand gestures. So the instruction is given on the computer monitor in front of the patient. And here you can see actually cortiQ running, where we are doing the high gamma mapping of the most important locations. And here I stop a little bit because it's super interesting. So what you can see here is an activation of the visual cortex because the person is seeing the hand. And then we find, of course, also the sensory motor activation over the sensory motor cortex. But at this time, we just didn't understand what the bubbles here mean. So we thought this must be an artifact on these few electrodes that we had inserted here. And later on, we figured out that this is actually the shape and color sensitive region and also the phase sensitive region of the brain. And people at this time just didn't know the meaning of these red bubbles. So nowadays we are doing a lot of research to investigate the temporal base. And you will hear more about these experiments. So the phase region, for example, is located approximately here. And neurosurgeons just removed tumors or sources of epilepsy if they were located here. And this had the consequence that people didn't recognize, for example, family members anymore. So nowadays with the cortiQ mapping, we can pinpoint these locations and we don't remove them anymore. Not me personally, our research partners like Dr. Kamada. On the left-hand side, you can see how clean the ECoG recordings are. Very beautiful signal. In this case, we are visualizing 64 channels. Then you can just see how the red bubbles develop, but we will explain this a little bit more in detail. So we started to build an embodiment station, and here the idea was to put an ECoG electrode or a stereo EEG electrodes into the brain. Then we use our g.HEIM to record all the high gamma activation, but also alpha, beta, and other oscillations. This was all fed into our real-time processing system, g.HISYS Professional, that we already used many times during spring school. This environment was also able to control intracranial stimulators to produce audio stimulation, haptic stimulation, electrical stimulations of the periphery, and we were also to record, for example, with microphones or with eye trackers, body movements. And the idea was to ask the patient to do different things, and then we are recording here to learn where these things are represented in the cortex and vice versa. We could also stimulate, for example, the hand, and then we could see how the brain is activating. But we have also the intracranial stimulator so that we can basically stimulate every single pixel of the brain. Then we can observe what's happening. And here's an example. In this case, we stimulated 4 fingers, pinky finger, ring finger, index finger, and so on for a few seconds. And at the same time, we did the high gamma mapping. And then you can see here the red bubbles. So these 2 electrodes are responsible for the middle finger sensation. Here's the ring finger, here's the thumb, here's the index finger. So after a few seconds, we have actually identified the sensation area of the fingers. And this is something that we can do with this embodiment station. This is a video that Kai recorded many years ago while he's just shaking the hands. And this was pretty unique at this time because you could just see how quickly the high gamma information decodes this information. So beforehand, this experiment needed many, many repetitions. And then it was more or less one handshake, and we could actually map it on the brain very nicely. That's the big advantage of ECoG. And then Gerwin Schalk with Peter Branner developed the Siegfried technology. So in this case, you can see an epilepsy patient is solving Rubik's Cube. They are recording here the ECoG signal, and the red bubbles decode the high gamma activation over certain cortical regions. You can see about 4 electrodes are decoding, in this case, the finger region. Then Gerf was asking the patient to stop and relax to get baseline high gamma activation. The next task is to stick out the tongue. Just controlling if I'm sharing computer sound. I think I didn't do it. So let's start again. So the patient is now sticking in and out the tongue. You might see it in the video. You can see the bubbles indicate the tongue region after a few seconds. Then the person is supposed to do a kissing movement for a few seconds. You can see again the red bubbles reach a certain state after a few seconds, very quick. It's difficult to see the tongue or the kissing movement, but she's just moving the lips. And the last task is to listen to a story. This is activating the auditory cortex and Wernicke's area, which is responsible to recognize voice and to understand what somebody is saying. And you will see it, how quick
Mr.
this was.
Jones had to go to New York. He decided to take a train. His wife drove him to the station, but on the way they had a flat tire. However, they arrived at the station just in time for him to catch the train.
You can see after a few seconds auditory cortex and Wernicke's area is mapped, and if we compared with the mapping here, it makes completely sense. This is auditory cortex. This is sensory motor cortex. Kissing must be very close to the tongue region and also close to the finger region. So this is something that GERF did 2010, so 14 years ago. Then we produced our cortiQ system based on this development. And this is an early version of cortiQ that we developed. So on the left-hand side, you can see in this case 64 channels. The patient in Japan gets the instruction to relax, to record high gamma activation. So we designed a montage creator to copy in these different electrode grids that we are using. And now the person is looking at the image. This is super interesting because first of all, we get an activation of the face area on basically the bottom of the brain. And a few moments later, the bubbles appear on the tongue region of the sensory motor cortex. This is something that I mentioned before. Many years ago, we didn't understand the meaning of these red bubbles. Nowadays, we know.
あるところにおじいさんとおばあさんが住んでいました。2人には子供がいなかったので、おじいさんとおばあさんは神様にお願いしました。
So also here the temporal dynamics are super interesting. First of all, here the temporal base is activating. This is decoding shapes of objects, and he has seen the loudspeaker symbol. And then we get auditory cortex and Wernicke's area. This is again the shape region. This is the color region on the temporal base. And then we get an activation of the finger region. So you can just very nicely observe how different brain centers are activated. And this is very useful to understand these networks, especially also for neurosurgeons, because they can plan this, the surgery much better. Now we are coming to the reason for high gamma mapping. In the video, you can see the clinical standard with electrical cortical stimulation, like it's done nowadays in all the hospitals. So here we have again an epilepsy patient, and Dr. Kamada is, maybe I switch the sound off, Dr. Kamada is probing all the different ECoG channels against each other. So this means at the moment he is stimulating electrodes 11 versus 16 with electrical current stimulation, and he's slowly turning up the current from 1 milliamp to 2 milliamp, 4 milliamp, 8 milliamp, and so on, all the way to 10 milliamps. And then he's observing what the patient can feel, or if a finger is moving, whatever. And this is the mapping procedure which is done all around the world. So it takes 1 or 2 days to test the whole cortex and to get the detailed information. So you can see that she's reporting about the index finger, the middle finger. Now he's stimulating pairs 16 versus 12. And she's reporting that she could feel it in the fingertips. 17 to 18 is producing nothing. So then Dr. Kamada writes down no function. Here he produced a mouth movement. Then he's writing down this is the mouth region. See, she's indicating lips here. Now he writes down 12, 13 lip region. In this case, Sanadi is connecting the stimulation plugs to the electrical stimulator. So this is done at this time manually. This is picture naming. So the patient has to name the different pictures that she sees. This is for testing expressive speech region, so Broca's area. And they are trying to interrupt Broca's area. And when she's not able to do it anymore, they know they were hitting receptive language region. And this is what you want to avoid. Sometimes the current is turned up too much, and then the patient gets a seizure. This is, of course, something that you want to avoid because you have to stop the mapping procedure. And of course, it's also not good for the patient if you produce seizures. And this is just happening with electrical cortical stimulation. And is one of the reasons why high gamma mapping is so important. Because we are just sensing from the brain, but we are not injecting electrical current. For that reason, it's much safer. And here you can also see another application of high gamma mapping. Dr. Kamada used it, for example, to see how a seizure is propagating. So in the middle, you can see the ECoG recording from the patient. You can see the patient is sleeping, but the seizure is actually starting. And here we are showing the high gamma filtered signal. And then we are doing the statistical analysis with the red bubbles. You can see in the center here of the image, the seizure starts. Then it's spreading out. So in the ECoG signal, we can see it. In the high gamma mapping signal, we can very nicely see it. And then it spreads out to the whole cortex very rapidly. You can also see the patient is moving a lot. So with this tool, we can map seizure pathways very, very nicely. And this is also super interesting for neurosurgeons because they can learn how they can maybe interrupt the seizure pathway by cutting out or interrupting certain pathways. Now you can see the seizure is all over in the ECoG signal and also in the high gamma mapping signal. And it takes some time to come down again. With this tool, it's just much easier to understand how a seizure spreads out compared to only an ECoG recording signal that hospitals normally use. Now you can see it gets more silent. Patient is stabilized. And so on. This is how the cortiQ system actually looks like for the high gamma mapping. So we have a computer doing everything. This is the screen for the patient, where the epilepsy patient or the tumor patient is looking at. Then we have a G.tec with different head boxes where you can plug in the ECoG electrodes. So this is a 256 channels unit, which is good enough for most of the mappings, but you can also stack it to 1024 channels if you need a larger area to be covered. And then we work together with Tony Ridaccio from Albany Medical College in the USA, in New York. And together with Ridaccio, we developed this paradigm. So we are asking the person to relax for 15 seconds. Then the person should solve Rubik's Cube to get the finger region. Then the person should stick out the tongue to get the tongue region. He has to listen to an English story to get auditory cortex and Wernicke's area. And at the end, the kissing movement should be done to get the lips region. And these are very important centers of the brain. And the neurosurgeon learns actually what he should not cut out. This also allows to do the surgery more precisely. And recently there is a very nice publication from a group in Oregon. They showed that tumor patients actually live longer if they are cortiQ'd, because this allows a neurosurgeon to operate more precisely and they can remove more of the tumor tissue. That's the reason why patients are living longer. So this is a very important outcome, of course. And then we worked also together with Dr. Kamada. And the Kamada paradigm includes solving Rubik's Cube for finger region, reading a story in Japanese language, of course, so that we get reading. Then a person has to do mental arithmetic to get the mathematical center, very important in Japan, of course. Then the person has to listen to a Japanese story in this case. This is the relaxation symbol for getting baseline activity. And this is an example of the Ridaccio paradigm when it was used at Yale University in the US. And here you can see they implanted the 64 channel grid. This is indicating the hand region. So in this case, a couple of electrodes are responsible for the finger movements. This is the kissing movement. So the kissing region is very close to the finger region, makes sense. The tongue region is even closer to kissing, makes also sense. Lips are right beside the tongue. And listening is located on the auditory cortex as expected. And this is the information actually that comes out of cortiQ, and this is handed over to the surgery team. And then they know they should not remove this region here because the patient couldn't kiss anymore. This would be, of course, a nightmare. Of course, we did also a lot of validation studies. So we looked, for example, at fMRI and ECoG. So we were asking a person to be at rest, and then the person did finger tapping for 20 seconds. We did a couple of repetitions, and here you can see the fMRI bold signal. So this brain region gets activated when you're doing this in the scanner. And then we did it also with ECoG. We used the cortiQ high gamma mapping tool, and you can see the red bubbles are overlaying the bold signal just very nicely. This is, of course, exactly what we want to have. And if we are mapping receptive and expressive language, the language is, of course, also very important for everybody, then people are doing a picture naming task. This means you are showing a certain picture to the patient. The patient has to tell you the word, what he's seeing. If we look again at the temporal dynamics, then first of all, we get an activation of the visual cortex because the patient is seeing it. And a few moments later, Broca's area is activated because the patient has to remember the name of the picture and has to say it. And this is expressive language area, or also called Broca's area. And people are also asked to listen to a Japanese story, for example. So in this case, we get auditory cortex because the person is hearing something. And then we get Wernicke's area, also called receptive language area, because they are understanding what they are hearing. And finally, Broca's area is activated because they are giving an answer to the question that was asked. So this is very nicely represented with the high gamma mapping. And of course, it's also important that the mapping results are stable. So we did the same experiment just a couple of times in a row. And you can see for the reading, we get very consistent activation here. The red bubbles are on the same location. And also for listening, we get very consistent activation of the bubbles. And this is, of course, super important that the high gamma mapping is consistent. And then we compared it also with electrical cortical stimulation. So Peter Branner will talk a lot about that in his keynote lecture today. But here you can see that the neurosurgeon or neurologist is always stimulating pairs of electrodes. And then when he is stimulating, for example, these two ECoG electrodes, then he is marking them in red. So the current flows actually from one electrode to the other one, and somewhere between these two electrodes, the cells are stimulating. And in this case, it was identified as picture naming. This means when the neurosurgeon turned up the current here to 8 milliamps, then the patient could not name the picture anymore. And this identifies the region as Broca's area, expressive language area. The same happened between these two electrodes and also here and here. So wherever there is this red color, the surgeon was able to interrupt the naming in the picture naming task. And for yellow, it's actually basically the same. It's also picture naming. For the white electrodes, you can see there was no symptom, so the patient didn't feel or respond. And the gray ones are responsible for the auditory illusion in this case. And if we compare it with the high gamma mapping, then you can see we find very nicely Broca's area. So it's exactly the same location like with the electrical cortical stimulation, but here we need 10 seconds to get it. Here we need a couple of hours to get the image. And there's also a nice side effect. So we are also getting visual cortex because first of all, the person is seeing it. This is something that you cannot do in such a mapping procedure because you can only stimulate one pair of electrode and you are blind for all the other electrodes at the same time. So mapping temporal dynamics with ECS is like impossible. With high gamma mapping, it's just a very nice side effect, which is of course also very interesting for clinicians, but also for researchers. Yeah, cortiQ is already used in many countries of the world. And here's a slide from Nuri Firat-Inch. He was also one of the keynote speakers on the second day. And he worked together with cortiQ from Freiburg, a German company producing ECoG grids. They designed for him a micro-macro grid, which has had 8 by 8 macro electrodes that you can see here in dark blue. So this is the clinical standard. They had 1 centimeter spacing. And in between, they put much smaller electrodes. You can see it a little bit. So to record with a much higher spatial resolution. And then Nuri put the data glove onto one of the patients, had also a high resolution camera, and he was sending everything into g.tec professional to record the ECoG data, but also the motion data at the same time. And here you can see that he analyzed the high gamma frequency band in the standard band between 60 and 280 Hertz. This is the range that most of the people are doing. Here you can nicely see how the high gamma signal looks like when the person is doing a hand movement task. It lasts about 600 milliseconds. We see the activation from like 40 Hertz to 250 Hertz, something like that. Then he mapped it with the green color here on the ECoG grid. So here you can see where the hand movement produced activation. So just a few electrodes were active, but then he looked also at the ultra high gamma range between 300 and 800 Hertz. This is a frequency band that most of the clinical recording systems cannot capture because the filter setting and the sampling frequency just doesn't allow to sample high enough, and they are cutting off very often already at 70 Hertz. So even the high gamma mapping is difficult in some clinical systems, and the ultra high gamma mapping is even much more difficult. But then Nuri found a very interesting fact. So if he looks all the way up to 1 kilohertz, then you can actually see that the high gamma activation also reaches these very high frequencies. And then he mapped the ultra high gamma from 300 to 800 Hertz in red. You can see there's another cortical region responsible for the ultra high gamma. And this is super interesting because up to this study, people thought high gamma ends at 200, 300 Hertz, but Nuri showed it goes all the way up to 1 kilohertz. And this was possible because of our g.tec system, because we are sampling the data with 614 kilohertz. So we are doing a dramatically high oversampling. And then we have a digital signal processor inside, which does the oversampling to increase the signal to noise ratio, which corresponds to the oversampling factor. So we can get rid of noise with these oversampling techniques very nicely. That's the reason why we can record all the way to 1 kilohertz and we can still see the ECoG signal. Otherwise, it would just be hidden in the amplifier noise. And then we are also using 24-bit ADC resolution. This is also super important to be able to record these very tiny signals. And we have also 2 additional co-processors inside for a very ultra steep anti-aliasing filter. And this is increasing the signal to noise ratio again. So altogether we have 4 processors inside the g.tec to do all these tasks, and at the end we get a much better noise floor and we can see these signals. So like mentioned before, this ultra high gamma is not hidden in the noise floor. This enables us also to do cortical evoked potentials. So Dr. Kamada will also speak about them. These CSEPs are very small and they could also be hidden in the noise floor. And with our high-end technology, we can actually get also these guys. And this leads me to the explanation of CSEPs. So cortical evoked potentials were firstly described by Riki Matsumoto, 2004. So this was part of his PhD. And we are also very happy to have him here today to explain us the history, the current research that he's doing. But in this figure, for example, he identified Broca's area, expressive language region, and then he's stimulating Broca's area with a 1 Hertz electrical stimulation in order to find evoked potentials on other cortical regions. So here you can see in red color where he is applying the 1 Hertz electrical stimulation. And then he had 1 grid over the sensory motor cortex and another ECoG grid over the auditory cortex. And while he is stimulating here, we find actually evoked potentials over the sensory motor cortex, and that's called a CSEP. This means it has to do something with the language network. Then we are moving to the other grid, which is over the auditory cortex. And here you can also see evoked potentials on some of the channels. And with this very elegant technique, we can map a whole cortical network. So for language, you need the auditory cortex to hear something. These channels who show the evoked potentials are indicating that. Then you need Wernicke's area to understand what you are hearing. Then you need Broca's area, which is here to give the answer. And finally, the sensory motor cortex is activating to say the words. So with this technique, we get it in one single shot, the whole cortical network. And this is just a very nice technique for research, but also for clinicians. And here you can see some of our collaboration partners. So we work together with many hospitals, neurosurgeons, and researchers for analyzing these data sets. This is an experiment that we did with sensory mapping. So in this case, we put the biprotactile stimulators onto 5 fingers. We stimulate it for a few seconds, and then we try to find the sensation cortical areas responsible for the fingers. And in another setting, we put on a data cloth, and then we try to find the motoric centers on the sensory motor cortex for single fingers. There's some analysis, but this is the result. So for the motor mapping, you can see here the results. You can also see that we used a high resolution grid where the electrodes were closer together to get a better spatial distribution. This is how the fingers actually look on one patient as an example. So the thumb is located here, basically decoded by 2 electrodes. Index finger is here. It's of course very close to the other finger. Middle finger is here. Ring finger is here. Little finger is here. And this is the information that you would need, for example, to decode single finger movements in a robotic avatar. And for the sensory mapping, so if we stick something into the finger or if we vibrate the fingers, you can see the sensation of the thumb is located here. Index finger is here, middle finger is here, ring finger is here, and little finger is here. This is a big advantage of the high resolution ECoG grid that we are using here because we get enough spatial resolution. The clinical standard would have 1 electrode here and 1 electrode here, so we wouldn't see the activation so nicely like plotted here. This is sensory mapping in another patient. You can see here the thumb looked like that, index finger like that, middle finger like that, ring finger, little finger. So it looks pretty much distinct for every single patient. That's the reason why you have to do the mapping first to figure out where the locations are. And ECoG is just perfect for that. So you wouldn't get these precise information, for example, with fMRI. This is an experiment for somatosensory evoked potentials. So in this case, we did a median nerve stimulation. So we had just an electrical stimulator here on the wrist. And if you place the electrodes correctly, then you actually find the finger movement whenever you stimulate the median nerve. And this is used to identify the central sulcus. So this is the border between the motor cortex and the sensory cortex, and it's defined by the phase reversal. So on one side, you can see the potential goes up, it's positive. And on the other side of the central sulcus, it goes down, it's negative. And this threshold is termed CS or central sulcus. And it's a very important landmark for neurosurgeons because they find it first and then they know where they should start with the surgery. And with our high cortiQ module, we can actually map the central sulcus very, very quick. You can see 1 stimulation, 2nd stimulation, and we already have it. So in blue, you can see here the positive wave. In red, you can see the negative wave. And the border between the positive and the negative wave actually identifies the central sulcus, which is here. So you can see here the blue bubbles and the red bubbles. And just in between is the CS. And if you look at the MRI image below, then you can see it follows exactly this little landmark in the MRI. So I can replay it again. Look at the bubbles, how quick we can actually find the central sulcus. 2 stimuli, 3, 4, and we already have it. So it takes a few moments and it's mapped. So with ECoG, we can achieve a fantastic speed for doing these tests. And here's an example with a high resolution grid for a brain-computer interface experiment where we had the goal to control a robotic avatar. So to give a whole robotic human being back to a locked-in patient or a paralyzed person. This is a very nice experiment. So the person is supposed to do exactly what's on the screen, like leave the finger straight, and the brain-computer interface controls the robotic hand. So she makes different gestures. I'm showing you the video here with real movements so that you can actually understand what she is doing. Now she's making the peace sign. Also the robotic hand makes the peace sign. We can even decode the ECoG faster than the robotic hand is reacting. So you can see there are no false positives. The robotic hand is doing exactly what she is doing. There's one very interesting moment at second 28. You have to watch careful. So the next one will be a mistake. Watch careful. The instruction is peace. She makes a fist, and also the robotic hand makes a fist. Maybe I'll play it again. So the next one is the mistake. She makes a fist, the robotic hand makes the fist. This is actually perfect embodiment that we want to achieve with the brain-computer interface. So the robotic system should really do what the brain suggests. But this shows also that patients are sometimes making mistakes. And that's the reason why you cannot always expect a brain-computer interface to deliver 100% accuracy. But we want to have, of course, to have perfect embodiment for robotic system control. This is something that we also published in Frontiers for Young Minds, like also for MindBeagle. So this is a journal publication for children. It's also reviewed by children. So it's a very nice read up for non-experts to dive into the topic of cortiQ and high gamma mapping. And this is my last slide. So I would like to invite everybody to submit their brain-computer interface project to the annual BCI award. So we are doing it for more than 10 years. And the submission deadline is in 1st of September 2024. All what we need is a 2-page description of your brain-computer interface project and a video where you're explaining it. And then we have an international jury looking at all the submissions, and then you can get an award certificate and also some cash money. But of course, it's very nice to be part of it. And it's also very useful to have it in the CV that you are a BCI award winner. So everybody is invited. And with that, I'm ending my presentation.